disclosure, Alex was both my undergraduate professor and my PhD supervisor. And just a little mini bio on Alex. Lots of you know him, but let's just remind ourselves of his history. Alex started life, let's forgive him, by studying for a BSc in zoology, which he graduated from Edinburgh University. He then found sense and switched over to physics and earned his PhD in applied physics from the University of Strathclyde, which is in Glasgow in Scotland. From there, Alex worked through his career, and this year he retired as full professor from the University of Strathclyde. And I was working out last night. I've actually known Alex for 25 years. This is Suzanne as an undergraduate, probably about 1991, something like that. 16, first year at university, head up bum. No idea what life was about. So I was let's just say I was an impressionable undergraduate student, and didn't I have a fine array of clothes? Look at my clown pants and my manga t-shirt. Anyway, so I've known Alex for about 25 years. We've been on lots of cruises together, we've had lots of fights, we've drank lots of beers, and now we're friends, I think. It's safe to say. So far, he says. So, Alex has actually done a lot for the field of ocean optics. We were a small group in a physics department, so we're always outnumbered by just straight physicists. So Alex used to turn up at lecture time and deliver absolutely fantastic lectures. Alex is a very, very good teacher, a very, very entertaining communicator, a very engaging speaker. So he would give us our classes on biooptics, and we were all, as students, eternally grateful for a lecturer that, one, didn't talk about quantum mechanics all the time and make us learn lots of equations, and didn't want to make us stab our eyeballs out, and could write legibly on the board and in comprehensible sentences with punctuation. It was really quite refreshing. So Alex would turn up with his wonderful ring binder of notes, transcribe it all beautifully in nice cursive onto the blackboard, which we transcribed into our notebooks. And then he would wax lyrical about the absolute poetry of the octopus eye, and how it was much superior to our eyes, and indeed vertebrate eyes, and that lesson has stuck with me today. I remember being quite agog about all of that. So, as Kurt told us last night, we all just gave up on quantum mechanics at that point and said, to hell with that, this is far more interesting, let's all do biooptics, or at least a portion of us. So just to finish off, here's just a little montage of some of Alex's former and current students, so I was one of them. This is Portland last year, where we had a photo booth with lots of famous figures from ocean optics history, and we discovered that Alex really bears quite a remarkable resemblance to Angelo Secchi. And Catherine Mitchell very kindly provided me with a picture last night. I think Alex is here, looking very handsome, suave. Here's David McKee. David McKee and I went right through all our undergraduate and postgraduate programs together. And here's Catherine, who's now at Bigelow and Jacopo, that's just I'm studying with David, and of course Ina right there as well, who's also still at Strathclyde. We have another colleague, we don't have photos of him, he ended up in Google. So without further ado, I'd like to first of all thank Alex for agreeing to be our plenary speaker. I insisted that I got to introduce you, and Alex had a kind of catchphrase. Oh, not yet, put it back. Alex had a catchphrase. He would listen to all of our angst-ridden conversations about what the hell we were going to do with our lives, and he would offer us some advice. And I find myself actually saying it to my own students now. I have been Cunninghamized, and it was, get on with it. So Alex, get on with it, and thank you so much.
I'd like to thank Suzanne for that very warm introduction, and thank the committee for the invitation to give this talk, and thank all of you for turning up this morning. this sort of image and asking questions about what the nature of the materials is in the water and how much of the stuff is present, and we worry about this. And it is, I'm trying to persuade you, only one way of looking at remote sensing observations, particularly in shelf seas and optically complex coastal waters. There's another set of questions you can ask. You can wonder if these changes in remote sensing reflectance patterns can tell you anything about the processes which are responsible for the presence of the materials which we're so used to studying. So this is the same image, and you can, instead of saying, yes, a wonderful coccolithophore bloom, you can say, I wonder why that's there. How long has it been there for? What caused it? What does that tell us about coastal ecosystems, shelf sea ecosystems, and the factors which drive them? And there are two things, I think, which currently make those questions answerable. The first thing is we've now got really quite long time series of satellite observations, so you can distinguish the instantaneous event from the annual or the decadal pattern. And the second thing is that the shelf sea numerical modeling community has made spectacular advances in terms of the accuracy of its physical models, and they're now making inroads into biogeochemical models as well. And it seems to me that there's an obvious complementarity between the sort of synoptic surface views that we can provide and the three-dimensional understanding of processes which you can get from models. And we really should have a wider bandwidth of communication, I think, with our numerical modeling colleagues. If, however, we want to contribute to that debate in a constructive way, we need a strategy. And it seems to me that central to the whole thing is the presence of suspended particles in the water column. And if you think about suspended particles, then they are responding to physical drivers, and they are generating distinctive remote sensing reflectance signals. And so this is one of these classical forward and inverse arguments. We need ways of getting a good inversion to extract information. I'm going to talk, and some of you will start yawning at this point, about the Irish Sea, a favourite haunting ground of mine. And it's a good place to work because there are lots of observations, not just from our group. It's an extremely well-studied area, and there are good numerical models of the Irish Sea. Against it, there are difficulties of sampling. Nobody ever goes there in winter. It's just too miserable. And the frequency of cloud cover is such that you really have to work hard to see the signal through the cloud obscurations, which is the significance of assembling long data series. Most of the stuff that I'm going to talk about is based on a set of cruises which cover quite a lot of the Irish Sea. Those of you sitting in the front row might see a somewhat younger David McKee directing operations on the deck of the ship there. So we've got lots of observations, and these observations will be usual suites of radiometry and filtered pads and all the stuff that you know about. And also, there is a good hydrodynamic model of this region. It's continuously evolving, so the stuff I'm going to show you is old, and it's got better grids and better parameterizations and things nowadays. But the how do you get out of that? The central thing is this 3D bioclinic model. It's embedded within a much bigger model of the Atlantic, so that sets the boundary conditions at the edges. It does the usual river inputs, meteorological forcing, and optionally you can couple it to a variety of ecological models once you've got your physical outputs. So if we're going to think about this in terms of particles and particle dynamics, the first question is something that we're all familiar with, which is what is the relationship between the particulate population in the water column and the reflectance which we measure. And the key to the link to that, of course, is inherent optical properties. Probably the most useful way of looking at this, certainly from the point of view of what we're talking about now, is the Gordon relationship. There is nothing magic about this. It's simply a second-order polynomial fit to a lot of Monte Carlo calculations. So in the mathematical sense, it's not an analytical expression. It's a condensation, an empirical interpretation of what reasonable radiative transfer calculations tell you. But because it's just a polynomial fit, you have to be very careful about your choice of the two constants, G0 and G1, otherwise the thing just goes and does silly things. Fortunately, Jean-Ping Li suggested a set of a G1 and G2 values for the coastal region, and it works rather well. So here we have the Gordon expression. The filled circles are hydrolite calculations which follow right on the Gordon model, and all these spots represent in situ observations. It's working pretty well until you get to this cluster, but actually they are coming from the highly turbid Severn estuary, and I think it's very difficult, you know, scattering corrections, all that sort of stuff, shallow depths and big instruments. So I'm not terribly worried about the fact that we're getting poor correspondences at high backscattering values. Have I missed anything? No. So once we've decided that we're going to follow that path, there are really two options. You can either, because you've got a BB over A problem, you haven't, inverting the Gordon equation doesn't give you a backscattering or an absorption coefficient, it gives you a ratio, and ratios can be quite awkward things. So you've got two options. Number one is go to an area where the absorption is relatively constant. For example, in the red, where seawater is a strong absorber, and then your signals are mainly generated by backscattering. And that leads you to far red, near infrared, single band algorithms, and Kevin Roddick's group's done some really nice work on the applicability of these things in shelf seas. The alternative is to go over to the dark side and start reading papers by Jean-Ping Li about the quasi-analytical algorithm and its variants and its modifications. And I'll get back to that, but basically it turns out to be quite a useful tool, provided you keep track of what it's doing and provided you're very confident on your validations. But to validate either of these things, we need a set of SIMPs, scattering cross-sections, absorption cross-sections. These may not be state-of-the-art, the ones I'm showing you, but they're actually physically self-consistent and realistic, and we found them quite useful as a starting point for doing reasonable optical closure in the Irish Sea and adjacent waters. For these SIMPs, it's not hard to work out that the responsive remote sensing reflectance to changing concentrations in what I'm calling MSS, mineral suspended solids, the response is always curved. You always finish up with a saturation value, but in the red wavelengths, here, the saturation value is way over here, and in the sort of concentration ranges that I'm going to be talking about, it's not quite linear, but it's a good, monotonic, sensible response. If you introduce CDOM, or if you introduce phytoplankton cells, of course, you mess up that relationship. But again, the thing, because it's working in the far red, is relatively insensitive to CDOM concentrations, and this is a set of chlorophyll ranges, a set of chlorophyll concentrations which exceed what we would normally see in the Irish Sea. And in fact, one of the things that I must remember to mention, wrong again. One of the things I must remember to mention is that Irish Sea CDOM concentrations away from the major rivers are actually rather low and rather constant. That simplifies a lot of what I'm going to be saying next. What I was supposed to be saying next is that in spite of these complicating factors, you can get very reasonable estimates, first-order estimates, of mineral suspended solids in the Irish Sea. This is the results of a whole lot of modelling with some data put on top to show the modelling is not stupid, with error bars. There are things down here which we know are diatoms, which show up in our gravimetric filtered bed measurements, but which don't really contribute very much to scattering and are not, in our sense, mineral suspended solids. If you take these estimated 95% confidence intervals and you transfer them to this graph as the dotted lines, then you find that our modelling nicely encompasses a completely independent set of work done by Dave Bowers and his colleagues. And they said that for the Irish Sea, this is a relationship between remote sensing reflectance and suspended minerals, and it's this relationship that I will probably use insofar as I need to use it later on in the talk. So let's start with something easy. And something easy has to do with what happens in the shallow edges of the Irish Sea, where there are mud banks and sand banks and lots of wave action. In that situation, then we go to our numerical model and we calculate something called the maximum, over a tidal cycle, the maximum bed shear stress. And it's bed shear stress which resuspends sediment. And so here's a couple of points where I've plotted the cycles of both bed shear stress and remote sensing reflectance. And you can see the cycles are by no means the same. The relationships are non-linear and different. But nevertheless, both the bed shear stress and the reflectance, and by implication the particle loading in the water column, show some kind of annual cycle. If you do that for quite a lot of stations, or positions on your remote sensing image, and then you normalize everything and you fit sigmoidal curves to them, you can do some kind of estimation of where the threshold is at which the sediment lifts off the bed and distributes itself in the water column. And that gives you an estimate of what, now this is boring if you're an optical oceanographer, but it's vitally important if you're interested in mud, coastal erosion, sediment dynamics, all that sort of thing. And although I've got no site-specific validation for this technique gives you numbers between 1 and 2 newtons per square meter, and here's a set of tabulated values for the UK coastline, and Ipswich is an outlier, but Ipswich is an outlier in all sorts of ways. But otherwise, you can see there's quite a good relationship between the ranges we're picking up from our simple algorithm and the ranges which we're supposed to get. Right, making it a little bit more complicated, the Irish Sea in its deeper waters is a complicated place. It's a complicated place because it partially stratifies and partially mixes in summer. It's one of the classic locations for studies of seasonal front formation. And if you collect a whole lot of data, this is an 8-year modest time series. From here on, I'm drawing heavily on work which was done by Catherine Mitchell when she was at Strathclyde. So thanks, Catherine, for this. She doesn't know I'm doing this, by the way, so the blame is mine. But if you draw a line down in images and you plot an 8-year modest time series, you can see from the whole deeper part of the Irish Sea, there is a seasonal cycle. And that seasonal cycle varies in amplitude, and rather paradoxically, the amplitude is strongest down here, which is actually one of the deeper parts of the Irish Sea. So this is not just a question of shallow waters are turbid, deep waters are less turbid. Dave Bowers has a really elegant little couple of lines of genuine analysis. You know, this is not modelling, this is sitting down with slide rule and pencil and paper and quill pen and working it all out. And he gets the sort of coarse details of this cycle neatly encapsulated in a diagram like this, where he says there are 2 stirrers. There is one stirrer at the top, which is stirring the water column through wave input. There is one stirrer at the bottom, which is doing the same through the ebb and flow of the tide across the seabed. There is a varying degree of coupling between the 2 stirrers, depending on whether or not you've got a well-formed thermocline, and then his predictions aren't bad. You don't get a lot of suspended sediment in summer, and you get lots more in winter. But lots is, I must say here, that lots is a relative term. We're never getting more than about 5-6 milligrams per cubic meter, no, grams per cubic meter of suspended sediment. These things are optically significant, but in terms of the amount of material that's being suspended, it's really very small. This takes a little bit of explaining, but I find it quite amazing. These are totally independent graphs from two utterly different people working in different fields. This one is one of these funny hydrodynamic dimensionless numbers. And what it actually calculates is the extent to which the effects of oscillatory flow over the seabed propagates turbulence up the water column. So a value of 1 says that the water column is going to be fully mixed, and a value of not very much says that the seabed propagation doesn't turn over the water column. And this is a graph by Claire Neal, which is the average winter red reflectance. And you can see, I hope you can see, that in spite of the fact that the colourings are inverse, you get quite good correlations. So here and here, here and here, the red says the bed stirring isn't propagating up the water column, and you have low winter reflectancies. And here, the blue number says the whole thing is mixed top to bottom, mainly by the tides, and here you have higher winter reflectances. So there are connections between physical parameters and what we're seeing from space. But remember, well, I haven't said this yet, so I'll say it now. As the winds decrease in winter and the solar heating increases, then you start to see density differences developing between the top and bottom of the water column. Not across the whole Irish Sea, but regionally. And these high regional areas of delta sigma actually correspond to the onset of a thermal stratification process. So here we have sea surface, and that is captured, by the way, quite nicely in sea surface temperature images. So here we have a sea surface temperature image, and you can see that on the cooler side, in the middle, there is uniform density and fairly uniform density, fairly uniform temperature as well. And then down here, the surface temperature corresponds to the onset of some kind of seasonal thermocline. As the water column stratifies, then, and the Bowers picture shows you this, you lose the particles which are being kept in suspension in the surface layer. And so we get gradually a well-defined boundary forming between stratified waters and mixed form. This is a classic seasonal front. Up here on the mixed side of the front, density is constant top to bottom, and the backscattering signal, which is basically a qualitative indicator of particle loading, is uniform. When we move over to the stratified side of the front, then here's the stratification appearing as a lower density surface water, and you can see we're losing particles, and the particles are accumulating down here and don't get lifted back up across the thermocline. Yeah, that was just emphasizing the point. But it turns out that the rates of loss of particles differ as the stratification establishes itself. And so although everything finishes up at a rather minimal summer value, during the sort of February, March, April time, and again in autumn, you have definite differences in the average reflectance across the front. And if we move to just one day in May, then you can see here's the sea surface temperature indicating the front position, and here is the contrast and reflectance, which is accompanying that front development. So what we have is really quite an exquisite way of testing shelf sea models because it's quite easy to do what I'm showing here, which is, in the course of a month, hit a correspondence between the reflectance signal and the physical parameters. But what we can do is we can do that on a daily basis, on a weekly basis, depending on how many cloud problems we have in our area of interest, and that allows the modelers to refine the parameterization of the physics and to get these, to get the timing right. And modest reflectance, or reflectance in general, is a really good indicator of whether the physics has been properly parameterized. Right, moving on and making things a little more hazy and waving my hands a lot more, we get to the quasi-analytical algorithm. This is the sort of thing you either know because you've read all the papers or you don't want to know. But basically, it uses the Gordon expression at the core, and it introduces an initial empirical step here, so you get some kind of estimate of absorption, and then you use your estimate of absorption plus a guess about the wavelength dependence of the back. Sorry, you use your estimate of absorption to get the backscattering at a given wavelength. You use an assumed relationship between wavelength and backscattering to extrapolate the backscattering to other wavelengths, and then, having got the backscattering at other wavelengths, you bring in the Gordon expression again, and you get spectral absorption. There are lots of variations on this, and we have a slightly tuned, it's true to the spirit of the thing, we have a slightly tuned version that we use for the REC. And if you do a whole load of hydrolyte modeling, and you drop some in situ data on top of the model predictions, you find that things are not perfect, but they're good enough for the sort of semi-quantitative work that I'm talking about here. So we do tend rather to overestimate our absorptions and to underestimate our backscattering, but it's not bad, and it's useful. If we take those QAA recovered absorption and backscattering coefficients, then you can generate time series like this, and this is really just a sanity plot from a specific location in the Northern Irish Sea. And what we have is annual cycles in 488 nanometer reflectance, which correspond more or less to the annual cycles in RRS 667, which we discussed previously. And we have backscattering maxima, which is generally aligned with the reflectance peaks, and we're turning up absorption peaks in summer. And this is not an area where there is a big CDOM cycle, so these absorption peaks are probably being generated by phytoplankton. So there's something useful going on here. And I want to go on and bolt some other things on top of these basic quasi-analytical parameters. And the two things I'd like to bolt on are, first of all, an estimation of the diffuse attenuation coefficient, and this is an expression from Yongping Li, which we have incidentally validated for the REC. And this is, there are lots of different ways of getting from KD at 488 nanometers to U14 depth. I'm going to use one that I developed, but it's no better than 3 or 4 other papers which get very similar results. If we look, again, this is for our waters and for this wavelength. I mean, if this was just a simple automatic solves all problem, then a lot of us could just go home. But for our water and our wavelengths, then here we have recovered against measured KD and recovered against measured U14 depth, and it's really rather pleasing. But remember that these validations are based on in situ radiometry and in situ absorption and backscattering measurements. There's a whole layer of stuff to do with atmospheric correction, which I'm not going to get involved in. Just to emphasize the point, most people do log-log plots in this sort of area, and on a log-log scale, we've got really quite encouraging recoveries of the absorption coefficient at 488 nanometers, the backscattering coefficient, the diffuse attenuation coefficient, and the U14 depth. That means we can generate infinite quantities The use you make of this depends on your interests, but it also depends on the frequency with which you can get cloud-free images. We have huge difficulty getting cloud-free images, but for a single day, you can see all this stuff, and you can imagine if you're working in a kinder area, you can start to look at daily, weekly, monthly movements of stuff. You can look at trends. You can look at the modelers will love it. It's an amazingly useful way of getting from remote sensing reflectance into the shelf-seen biology and physics without worrying too much about whether or not your chlorophyll algorithm is working correctly. And of course, you can do things like you can build up baselines. So here is an 8-year average at different months of the euphotic depth contours for the Irish Sea. And with those baselines, you can then look for trends, you can look for events driven by storms, you can possibly look for perturbations owing to oil spills or dredging or whatever it is, or you can just hope that you're eventually going to see or maybe not see over the years some kind of evidence of climate change or local weather changes. I want to push this a little bit further because we've got 10 minutes left and because it's always tempting to speculate. And this is just the start, and I'm hoping that some people will take this up and do it properly. But if you write down the particulate absorption and backscattering coefficients in a slightly redundant fashion, and you introduce these two row parameters, which are the backscattering to absorption ratios for phytoplankton and mineral particles, then it doesn't take much algebra to separate out the contributions of mineral particles and chlorophyll to the total absorption coefficient. And if you do that, and you take all the data that we've ever been able to collect, and we plot each thing pixel by pixel, then what you find is that on a graph of backscattering against absorption, the Irish Sea draws you a triangle. And the boundaries of that triangle, more or less, are given by the ratios of the BB to A for phytoplankton and minerals. This is the phytoplankton one, this is the mineral ones. Although you can do this by fitting the lines to the cluster, these lines were drawn just using the SIOPs, which I showed you half an hour ago. And on this graph, although obviously you can mess around with the axes and do corrections, the offset here is because there is a small but relatively constant absorption effect going to seed on in the Irish Sea. Then if you break the thing down into seasons, and again because of our cloud difficulties, we're having to aggregate data for this, you can see that the Irish Sea moves around, as it were, within these boundaries. So in winter, your reflectances are all up here, and this is the whole shebang. Your reflectances are up here, high backscatter, low absorption, a lot of mineral particles in the water. Down here, low backscatter, high absorption, fewer mineral particles, although there's quite a lot because we're including all the edges and the estuaries and so on. But basically, the Irish Sea is moving around within this parameter space. And this is just a reality check. If you look at the proportion of the total absorption which we allocate to mineral particles using this partitioning method, then you find that, sure enough, in January, there are lots of mineral particles doing lots of absorption, and they just fade away gradually. You can see the front developing, and then you've got nothing but a few patches around the obvious. These are all explicable features, and then in October, the thing starts to pick up again. If we look at a time series rather than a map, then we begin to see that the suspended mineral part, or the fraction of absorption allocated to minerals, peaks in January, that the fraction allocated to phytoplankton peaks later in the year, again just for the station. And so we can start to talk about phytoplankton phenology and bloom timing and all that sort of thing. And then it gets interesting, because if you move around the IC, you find that phenology changes. Here, on the stratified side of what we've now decided is a good seasonal front, you get a double phytoplankton bloom, spring and autumn, and here the phytoplankton bloom is much less intense, sporadic, but this is just for 1 year. You can pull better signals out of this by aggregating multi-year data, but basically we have a little piffly bloom in the middle of the year up here, and we have 2 good spring and autumn blooms there. And if we look at the associated estimates of euphotic depth, the interesting thing is here, you can see the phytoplankton are actually affecting the euphotic depth quite significantly. Here, they're not. The euphotic depth goes up to its summer values, and the phytoplankton just take advantage of the light that's available. Now, if I wave my hand in some kind of deliberately vague way towards the phytoplankton people in the audience, biogeochemists, then maybe I can persuade you, and this is purely speculation, that you could begin to deconstruct this. You could start to think about areas of the shelf where it's light limitation which is determining bloom phenology, areas of the shelf where it is possibly nutrient limitation. This is work for the future. So the question is what we're going to do next. Here's a list of possibilities, but I think the take-home message is that it's very easy for us in ocean optics to get fully engaged in new satellite sensors, new algorithms, new in situ instrumentation, and forget that we've actually got quite a big contribution to make to the wider field of shelf sea science.